Hello everyone, it's Dr. Gutierrez. Today I'm going to talk about the digestive system. And one of the things that I will mention ahead of time is that most of the images I use are fair use. There are a lot from KenHub. There are some that I picked up along the way, which I don't remember where I got. So if someone knows where they're from, let me know. I can remove them if they're copyrighted. But when I talk about digestion, I actually want to start with what's called the gastrocephalic reflex, and that is pretty much what happens when you see, hear, smell food. Pretty much, it actually triggers a parasympathetics, and the parasympathetics will trigger the intestines to start moving food, to make way for food that's coming in. It also starts the salivary glands secreting the saliva. And so embryology, it's actually kind of nice to talk about because what we have is, if you remember, by week 3 we have three germ layers. These are your innermost layer, which is your endoderm, your outermost layer, which is your ectoderm, and the middle layer, which is your, I'll put the nerve tube in here too, which is your mesoderm. And part of the mesoderm will come and form a area around the gut tube and will actually lead to an opening inside a cavity here. And you will have a lining in there called the uh, peritoneum. And so the tubes are usually, the gut tube is usually a peritoneal organ. Retroperitoneal organs will actually be in the back, like the aorta and uh, the inferior vena cava, for instance. And so what ends up happening is this starts growing, and the gut tube will form in this ventral body cavity, and endodermal cells will actually form in this area to form the liver, actually the liver over here, and the spleen. And this will rotate around, so you'll have your liver kind of bound up in front and the back, and the spleen going in the front and the back, the spleen being to the left side on the back. So your liver will be in front of your gut tube to a degree, which we'll show you later. In the gut tube, we do have something that forms, which is called the respiratory bulb, which will form the trachea, the bronchus, the lungs, and there are some genetic defects of the respiratory tract and upper GI tract that will affect both systems. One of them is called a congenital esophageal atresia, which what happens is the esophagus forms a blind sac, so food goes into the esophagus food goes into the trachea instead of esophagus. So you do have a lot of issues with newborns vomiting when they eat. Now, one thing I actually do want you to know is the quadrants, and it's important to know the quadrants with the GI because this will tell you where certain things are. If you find the xiphoid process, we can make a line straight down through the umbilicus. We can also take the umbilicus and go across, and so we have 4 quadrants, right? You have one over here, which is your upper right, upper left, lower right. And so you can actually determine where certain things are here. You do have another one where you could go from the middle of the clavicle straight down, both sides, from the ribs across. And so you can divide the abdomen into the epigastric, the periumbilical, the pubic, and the flanks would be here, the hypochondriac, and the iliac here. And just showing you the lines. This is the other 2 that I was talking about. And so with that, we can actually start by looking at the mouth. And one of the things that's important to look at the mouth is the muscles of mastication, because they are going to help you chew. So you can actually open and close your mouth. I recommend going to the digestive list so you can see which ones are important. You also have glands. You can see a gland here, which is on the side, which will be called your parotid gland. You have a gland underneath the tongue, which is called the sublingual gland. And you have a gland underneath the mandible, submandibular gland. And these are going to produce slightly different types of salivary secretions. We also have teeth. And one of the things I actually will tell you is if you go into dentistry, they will actually number the teeth as well. But if you look at this one, it tends to have a flat area here, comes up, and if you were to turn it around, it would be kind of a sharp area here. So it looks almost like an hatchet, a axe. And these are your incisors. Next to it, you have these that have this nice sharp point. These are your canines, which are next to your incisors. And the incisors, if you remember how your bones and the mouth work, when your mouth opens and closes, it slides forward when it opens, slides back when it closes. So the incisors make a cutting-like motion. The canines kind of stab your food, and they help hold it in place so you can tear. I will skip these premolars and go to the molars, which what you'll notice is they have this nice broad shape, which allows you to grind food. The premolars kind of have a little of everything, and they do a slight variation of it. They do kind of cut a little better than you would expect, and you can tell what these teeth do based on animals that have predominant types of teeth like this. Now, over here you can also see other things. You can see the inside here is a cavity, which is called the pulp cavity. You then have the area over here called the dentin, and you have the enamel, the white area up here. We can also see it as the roots here, the neck, and the crown. And we'll talk about those here. So there are different parts of the teeth. We have the crown, which is the outer area, and it is made of enamel, which is the hardest substance in the body. The truth is, a lot of people tell you, oh, the hardest bone in the body is XYZ. The real hardest bones in the body are the teeth. They are made of the crown is, and the enamel are made of something called substantia adamantium. Then you have the dentin, which is over here. Now that is harder than most bones, but it's not as hard as enamel. You still have cells and living things here. The enamel does not actually have too many cells, so when it actually is broken, you do tend to have issues. And then you have the pulp, where you have the blood vessels, you have the nerve where you feel, and so that's actually the structure of the teeth. Now, underneath this, the crown, we have the neck, and that's actually usually found in the gum line. It is made of dentin here and pulp. And then you have the roots, which are attached to the mandible and maxilla in the alveolar processes via a gymphosis joint, and it uses something called cementin to hold it together. It also does have a periodontal ligament which holds this in place. The blood supplies for this will come from the roots. And so if the root is broken and the blood supply is broken, a lot of times you will lose the tooth. There are some great dentists that can put this together. There are different emergency people who can actually work if it does break here. But a lot of times when this is broken, you will end up losing the tooth. Now, what are the tissues found within the mouth? Here you can see you have a long structure of cells. The basement membrane would be down here. The lumen of the mouth would be here. And so you have a stratified epithelium. And if you look closely, you may be able to find that these cells are stratified. Are flat. So it's stratified squamous epithelium. The reason you don't have keratin is because the keratin will wash off with the saliva. So it's stratified squamous epithelium. And then we have the salivary glands, which I mentioned before. The parotid, the submandibular, and the sublingual. And these are going to produce three things. Mucus, which helps lubricate the food. Water, which helps dissolve food particles. And amylase, which, always remember, anytime you see ACE, it's an enzyme. And amylase is an enzyme that breaks starches into simple sugars. So in the mouth, you start breaking up starches into simple sugar. You do not get any absorption here yet because of the stratified squamous epithelium, but you can get some underneath the tongue, I should mention. Here, I think I got this from KenHub, but it just shows you the different glands. And one of the things that you'll notice is some of them are very clear, some of them are very dense. The dense ones are the parotid gland, which secrete mostly enzymes and water, and so they stain really well. Mucus doesn't stain so well, so you can actually see the sublingual gland has a lot of mucus, and the submandibular kind of is a mixture of both. The tissue in the glands is this one here. Now, if you think of this layer here, as the basement membrane, and you think of a hole, cavity here. This is one cell layer thick, and each cell is roughly, not completely, but roughly a cube shape. So it's simple cuboidal epithelium. So what happens in mouth? You take food in your mouth, the saliva mixes with the food as you chew, and then you swallow it and move it to the esophagus. The food you swallow is called the bolus. And the bolus goes down this tube called the esophagus here. And the esophagus ends at the stomach. And so you have a sphincter here called either the gastroesophageal sphincter, the cardiac sphincter. There's a few other names for it. But I actually tell my students to think of what name you like most. You will notice that in the stomach, you have this big curvature here called the greater curvature, this lesser curvature here called the lesser curvature, and this area here, which is next to the heart, so it's called the cardiac zone. The fundus is the body, and the pylorus, which I'll talk about later. One of the things to know is the lumen of the esophagus is mainly made of stratified squamous epithelium. You think about it, whatever you ate, whatever you chewed, you're still getting it to rub against the inside of the esophagus. So you want to have specialized tissue with that. This is one of those pictures I don't know where I got from, but it does show you the layers. So you have the mucosa, which is mainly the epithelial tissue, and the lamina propria. You do have a muscularis mucosa in there as well, which is just a smooth muscle layer of the mucosa. Then you have an area called the submucosa. Now the submucosa is just underneath the mucosa. And then you have the muscles, which are called the muscularis layer, which is a circular muscle and a longitudinal. The circular will actually help push things down. The longitudinal will constrict to allow the food bolus to be pretty much to go only in one direction. Past the muscularis, you have the serosa, which does have nerves and does have some of the glands that the blood vessels that are going to help maintain the esophagus. Now, the veins in here are pretty sensitive to pressure, and sometimes when someone has a major violent retching, it can tear the veins. It actually can cause the veins to become varicose, esophageal variceles, and they can tear and cause a life and death situation. Now, this is from KenHub. I know because I can see the KenHub written up here. I really love the site. I recommend everyone who wants to learn anatomy actually get a subscription for it. And you can see over here the lumen, which is just a hole. You can see the stratified squamous epithelium. You can see the mucosa underneath. Here, mucosa is the entire layer. Then you have the submucosa down to the mucosa. And then you have the muscularis layer, which you have your internal, circular, and longitudinal outside. And then you have your adventitia or your serosa. And then we get to the stomach. The stomach enters via the sphincter here. Again, you can name it, for me, you can name it either gastroesophageal sphincter or the cardiac sphincter. And it goes into the stomach, which is a big bag here. Now the stomach has a bunch of folds inside called rugae, which allow it to stretch, which is why you can see professional hot dog eaters not necessarily be the biggest guys, yet their stomach can grow and push down. Unlike the esophagus that has two layers, this has a third layer called which is deep more, which is called the oblique layer, which is running to the sides, running inside pretty much to deep everything in these different angles. Now, there is a sphincter at the end of the stomach called the pyloric sphincter. The area here, and the last part of the stomach, is called the pyloric region. And the pyloric sphincter keeps the stuff in the stomach from going out. Now, once things get into the stomach, it is hit with acid, and acid will start chemically breaking down food. This isn't enough to break down starches or proteins or fats, but it's enough to kill anything in there. In fact, the pH inside the stomach is about a pH of 2, which is extremely acidic. And when the bolus gets in here, it's no longer called a bolus, it's called chyme. Inside the stomach, again, you'll notice I love KenHub. You have surface mucosa, epithelial cells. You have mucosa cells, which are actually the goblet cells. And if you think about it, the respiratory tissue came from the GI, so it does make sense that you'll have the goblet cells. You have parietal cells, which make hydrochloric acid, and you have chief cells, which make pepsinogen. Pepsin, and chymotrypsin are proteolytic enzymes. If you think about the word, proteo means protein, lytic means breaking, enzymes. I did borrow again this slide from KenHub. They said it was okay, as long as I gave them credit every time I did. And this one's also from KenHub. You can actually see how you have these long projections here. You have simple columnar epithelium, by the way, and the mucosa, and the goblet cells produce a nice mucus layer here to keep the acid from breaking it apart. You can see where the cells that actually are going to produce the hydrochloric acid, the parietal cells, the chief cells, are going to be up here, and they're going to secrete into this gastric pit. And these areas here are called your gastric crypts. You do have a submucosa, you do have blood vessels going in there, but you don't have much absorption. About the only absorption you can get here is the non-steroidal anti-inflammatories like aspirin and Tylenol. You also can get caffeine and some alcohols that go through there. And this will go through the gastric veins to the hepatoportal veins and end up going to the liver first, where it can actually help detoxify. Over here you can also see the oblique muscles, the circular and longitudinal, and then the serosa underneath. You can see some Arbuck's plexuses here, which does tell the muscles to contract. Once you get past the stomach, you end up going into the small intestine. The first part is called the duodenum, which I'll show you later. If you look here, these areas here are the small intestine. The one over here are called the large because the diameter is a lot bigger. Jejunum is the first part. It's about 2.5 meters. Then you have the ileum, which is the last part, which is about 3.5 meters. Most of it will be the the duodenum is about 22 centimeters, and you have the jejunum, which is about 2.5 meters, and the ileum is the last part. Now, you are going to start absorbing here, and it is filled with bacteria. But these bacteria are really helpful because they make vitamins and help you break down any disaccharides you cannot. They will end up making gas, which is another issue. So when we get into it, we can see you do have goblet cells, and you have these finger-like projections, which are called villi, and they expand surface area so you can absorb. And you do have actually areas inside here with lymphatics, lacteal, and capillary beds, which will help absorb nutrients from the lumen. You do also have the muscularis layer, which do have the circular and longitudinal muscles again. Now, I did want to show you the duodenum over here. So the stomach is attached here, and you can see the gallbladder coming in here. You can see these two things which are coming from the liver, and you can see the pancreas here. Now, this is a pancreatic duct, common bile duct. This would be hepatic, and the cystic duct here. And whatever the liver secretes will go into this area and drop into the duodenum. Whatever is in the pancreas will be secreted into the duodenum through the pancreatic duct. And a lot of the things that are actually secreted here help in digestion, which I'll talk more about. For instance, in the pancreas, you do make a lipase, lip for lipid. ACE is an enzyme, so it's an enzyme that works on lipids. It breaks down fatty acids and triglycerides, takes fats and breaks them down in fatty acids and triglycerides. It also makes amylase. Again, amylase, we talked in the mouth, how it takes carbohydrates and breaks them down into sugars. It also does produce some bicarbonate, which will help neutralize the chyme that's coming in from the stomach. From the liver, we do make bile salts, which help emulsify fat. What I mean by emulsify is if you have a big glob of fat, it breaks it into smaller globs so lipase can work better. Now, as you're going to make more bile salts than you need, we're going to store some of them in the gallbladder, and whenever you need it, you can actually excrete it technically into the small intestine. So you don't really need a gallbladder, and if someone has stones, we can take them out. Now, I want to talk about the large intestine. Mainly, the thing that's happening in the large intestine is the absorption of water. What you will see is a small intestine, you'll see the large intestine, and you'll see that it goes from the small intestine up here. So we're going to call this part of the large intestine the ascending colon. The first part here is called the cecum. And between the ileum and the cecum, you have a valve called the ileocecal valve. You also can see a nice projection here called the vermiform appendix, which is what gets inflamed when someone has appendicitis. You do see these bulges here called haustra, and you see this line here called the tinea coli. Now, the ascending colon comes and it folds here, and the liver is over on this area here, so you cut a hepatic flexure. And then this food stuff goes to from the right side to the left towards the spleen, and it actually folds there, so we call this a transverse colon, and we have the splenic flexure. And then food goes down, so we now call this the descending colon, and then we have something that looks kind of like an S here. And so this structure here is called your sigmoid, and then you go to sigmoid rectum, and then you go to the rectum, then the anus, and the two anal sphincters. Now the two anal sphincters are thickening smooth muscle. The anus does have the stratified squamous epithelium, which is a little different than all the other aspects of the lower GI tract. Now I do want to talk a little bit about the accessory organs, which I touched on. The first one is the pancreas, which I mentioned makes bicarbonate. It does make amylase, not protease. This would be amylase. It makes lipase, and it secretes it into the small intestine. You do have proteases in the stomach, and you do also have proteases in what's called the brush border of the small intestine, which are going to break down the proteins. You also have specialized cells that, instead of excreting stuff, secrete it into these blood vessels here, which are your islets of Langerhans, which secrete insulin by the beta cells, glucagon by alpha cells, and somatostatin by beta cells. It does have an area up here, which is bigger, which we call the head, the tail here, and the body is between it. You can also see the gallbladder up here, which I'll mention in a little bit. Now, when we look at the pancreas, we see a pancreatic duct, which is going to go to the intestines. We also see, if we go deeper, we'll see this big island-like thing here. Try to highlight it a little bit. Again, this is a KenHub image, and that's called your islet of Langerhans, and you have your zymogenic molecules, zymogenic particles here. You do have acinar cells, and so these are the cells that are going to actually secrete the bicarbonate, some proteolytic enzymes. You're going to actually produce lipase, amylase, and that's going to go inside. Now, we also have the liver. Now, I'm looking at the back of the liver, and you can see this big ligament here called the ligamentum teres. You can see this area here called the falciform ligament, and you can see how you have different lobes. Now, this is a back, so I can tell you that this will be the right lobe. I'm sorry, the left lobe. This will be the right lobe, and you can see a little tail-like lobe here. The tail-like lobe is called the caudate lobe, and over here you can see a almost square-like lobe called the quadrate lobe. You can also see a vessel we've talked about before, the inferior vena cava. The hepatic veins drain into it. You can see the hepatoportal vein, hepatic artery, and the bile duct, and together these three are the hepatic triad. You'll see the hepatic triad in the tissues a little more. And so you can see how the liver is organized. The right lobe, the left lobe, right lobe here, left lobe here, caudate lobe, and quadrate lobe. It does make more sense to look at a three-dimensional model of the liver so you can see what I'm actually talking about. Now, when you look here, you can see that you have three vessels on one side, and you have a big vessel coming on the other side. The big vessel would actually be the hepatic vein over here, venule, and the three vessels here would be the hepatoportal vein, hepatic artery, and the common bile duct. And what usually happens is the hepatoportal vein, venule, will actually try to push fluid through the hepatocytes to the hepatic vein, which will go into the hepatoportal, to the central vein, which is actually part of the hepatic vein, which drains into the inferior vena cava, going back to the heart. So you can filter and destroy some of the stuff that shouldn't be there. For instance, if you eat a big sugar meal, it will go into the mesenteric veins, into the hepatoportal vein, and come into the liver. You'll also have blood from the spleen coming in here, which will end up giving you the unconjugated bilirubin, which the hepatocytes can conjugate and then send it to the central vein. And you can make the bile salts a lot of times using some breakdown products and send it to the common bile duct for it to go to the intestines to emulsify fat. And we do have the spleen, which is part of the lymphatic system, which I talked about. So it does tend to make bilirubin from old red blood cells. It sends it to the splenic vein, which will drain into the hepatoportal vein, which goes to the liver. And it is conjugated, and some of it is used to make bile salts, giving the yellowish color that will end up going into the intestines. Now, this is actually something that does go into physiology. As you're eating and you're bringing most of your amino acids to the liver, we mentioned, make most of the circulatory proteins. And one of the proteins it makes is a high density lipoprotein. Remember, lipo means fat. So a lipoprotein is a fat protein. And if it's high density, it means it has more proteins than lipids. A low density lipoprotein will have lower amounts of proteins than lipids, and so it has a lower density, so it floats. And then you have the very low density, which pretty much means you have a lot of lipids associated with your proteins. And what usually happens is your HDLs are made in your liver, and they're released through the hepatic veins to go to the right atrium eventually. They will end up scavenging for fatty acids and triglycerides, bind them, and when they do, they become low density lipoproteins, or LDL. That's why if you have a high LDL to low LDL ratio, it's healthy. It means that you're pulling fats out of your blood to move them on something else. If you have high LDLs compared to HDLs, it's a sign that you are going to have cardiovascular issues later. Now, the LDL will move the fatty acids and triglycerides to organs that need them, and they can actually be stored for energy as well. And so that's why the HDL-LDL ratios are used a lot of times as a clinical test. It does also show you how your liver works, and if your liver is damaged, a lot of times you will not necessarily make the amount of HDLs that you need, so you will have a higher LDL, which leads to more cardiovascular accident. So going back to the small intestines, all this stuff is going in there, and what you have is you have these villi and a microvilli, which end up giving you a brush border. We have anything water soluble that can come in here and go into the mesenteric veins. So the mesenteric arteries are coming into the villi and making capillaries. The mesenteric capillaries are taking the sugars and the amino acids. Remember, the amino acids the proteins are broken down into amino acids. The carbohydrates are broken down into simple sugars, and the fatty acids are broken down into the fats are broken down into fatty acids and triglycerides. But as proteins and sugars can go into blood, they'll go into the mesenteric veins. Fatty acids and triglycerides cannot really travel in blood alone, so they'll go into the lacteals, and that's actually how they get to the heart. One thing to remember is the fatty acids and triglycerides go to the heart first, and the reason that's important is the heart requires a lot of energy, and it can use oxygen to break down fat, and so that's one way it does help. So where do the nutrients go? Amino acids that enter the capillary beds go to the mesenteric veins. The mesenteric veins will end up going into the hepatoportal vein. The hepatoportal vein will go into the liver. Sugars will again go to the capillary beds, to the mesenteric veins, to the liver. Fatty acids and triglycerides will go into the lymphatics and go to the heart first. And the reason they're sent to the heart first is so your heart can use it as energy. Remember, if you have to go out and look for fat, it's actually a hard thing to do. Today we can actually get fats quite easy because we have fast food and processed foods and everything. So you'll notice that I've talked about the hepatoportal vein system quite a bit. The reason this is important is it takes amino acids and sugars straight to the liver. A lot of times the liver doesn't need insulin to pick up the sugars. It actually relies on the high concentration coming from a meal. And so this actually will keep the sugar from spiking too much in your blood. If you do have issues with your liver, a lot of times sugar levels will actually come up after a meal. Amino acids also go there, and again, those amino acids can be made to make all sorts of circulatory proteins, in particular high density lipoproteins, which will go into the inferior vena cava, into the heart, and the fatty acids and triglyceride go into the heart. The heart will use what it can, and whatever it can't will be picked up by the high density lipoproteins, converting them to low density lipoproteins. These are going to go into the lacteals, continue, and so these are actually, the fatty acids are taken into lacteals and going to the heart. Now, one thing to notice is I talked about water-soluble and fat-soluble aspects, how they come in. I talked about carbohydrates that you can break down, but there are some stuff you can't break down. And whatever you don't break down stays in your GI tract. And that's actually really important because it acts as a bulking agent. We talk about it as fiber. It is cellulose. It is plant-based carbohydrates we cannot break down. And they actually act as a bulking agent, so it's easier to defecate, easier to move stuff out. You will notice that there's very little that's actually sent out into the GI tract. Most of it is coming from the pancreas and liver. And so the thing is, a lot of times people talk about cleaning out your system by doing something to your intestines. Well, your intestines, the best way to clean it out is to eat high fiber meals. In fact, most people say we should all be eating higher fiber meals because of its protective nature to many diseases. I hope you enjoy this and have a nice day.